Welcome to the fourth episode of O-Levels Biology. I'm Larry, and today we'll be covering on Chapter 4, Enzymes. This is based on the latest science syllabus. This is a general view of this very chapter, and for more information about me and the services I provide, please visit www.larrylu.com. I hope this will be helpful to you. Please sit back and enjoy the video. Thank you. This is a picture of an enzyme and substrate. When enzyme and substrate comes and combine together, an enzyme-substrate complex will be formed and reaction will occur. Now, let's first touch on the definition in this chapter. Catalysts are substances that increases the rate of reaction, and in this case, they are called enzymes. Enzymes are biological catalysts which are essentially proteins. The active site located on an enzyme is the part which allows substrate to bind for reaction to occur, and is indicated by the white part here. Substrates are compound which enzymes act on and is indicated by this blue part here. In order for reaction to take place, activation energy is required and is defined as the minimum energy for reaction to occur. In order to fully appreciate these terms, we'll look closely at them later in this chapter. This is a graph showing an enzyme reaction. There is an obvious difference between the green and the red part. The original energy required for a reaction to occur is called the activation energy and is around this level. However, with the aid of an enzyme, the energy required will be lowered as reactant particles possess more energy than activation energy, and thus reaction occur at a much faster rate. In biochemistry, hydrolases are enzymes that catalyzes the hydrolysis of chemical bonds. It might be tough to remember all the hydrolases, but let's use the mnemonics method. For carbohydrates, we use CLAMS, CLAMS, cellulase, lactase, amylase, maltase, and sucrase. For proteins, PET, PEP, pepsin, erepsin, and trypsin. And lipase is, lipase. Now, you must be wondering, how on earth does the enzyme comes into contact with the substrate? It does so by using the lock and key hypothesis model. Imagine slotting your house key into the house lock and together they form an enzyme-substrate complex. It is highly efficient and highly specific. Well, if any key can fit into your door lock, I guess it's time to change your door lock. The other model is the induced fit hypothesis model, in which the enzymes come close to the substrate molecules, and the active site actually accommodates by slightly changing its shape to facilitate binding at the active site. Let's take a look at the effects of temperature on the enzymes. This is a graph of an enzyme-catalyzed reaction. As you can see from this graph, as temperature increases, the enzyme activity will increase as well. At around 37 degrees, the enzyme activity will reach its maximum. This rise in temperature results in an increase in kinetic energy and particles start to collide more with each other. Thus, the enzyme-substrate complex will be formed at a much faster rate. As temperature increases further, the enzyme activity will decrease rapidly and eventually the enzymes will be denatured. This essentially means that the enzyme have lost its function permanently. And this is why boiling temperature water are not advisable to be used together with detergent to wash clothes, as the enzymes in the detergent will be denatured and rendered useless. Other than temperature, enzyme activity can also be affected by changes in pH. The optimum pH for enzymes is around 8, and any deviation from this point will result in denaturation of enzyme. This is due to the pH disrupting the electrostatic charges, resulting in electrostatic repulsion, altering the enzyme 3D structure. Substrate and enzyme concentration. Now, suppose if substrate concentration has already reached its saturation point, adding more enzymes will not increase the rate of reaction as indicated by the horizontal line shown. This is due to the enzyme not having enough substrate molecules to act on. However, if the substrate is at a high enough concentration, adding more enzymes will lead to increasing rate of reaction as shown in the ascending line. So now let's do a very quick recap. Enzyme and substrate combines to form an enzyme-substrate complex. There are two types of model in which enzyme and substrate interacts, and they are the lock and key hypothesis and induced fit hypothesis model. Temperature and pH can affect enzymes. At temperature more than 60 degrees, enzymes will be denatured. And any deviation from optimum pH 8 will denature enzymes. The rate of reaction can be affected by both substrate concentration and enzyme concentration. Thank you for watching. Stay tuned to Chapter 5, Animal Nutrition. For more information about me and my services, please visit www.larrylou.com. See you and goodbye.